Hi, my research was aimed toward identifying a dietary supplement that can effectively regenerate myelin sheaths in zebrafish embryos as a model for multiple sclerosis. The purpose of this was to determine a way to lessen the demyelinating effects of this disease. Myelin sheaths are thick layers of proteins and fatty substances that wrap around the axons of nerve cells, acting as insulators that significantly help nerves in transmitting electrical impulses quickly and efficiently. The destruction of myelin is known as demyelination, which involves either damage to the myelin itself or to myelin-producing cells. The most common cause of demyelination is multiple sclerosis, or MS, which affects nearly 3 million people worldwide. This autoimmune disease, by causing the immune cells of the patient to attack its own oligodendrocytes, causes a wide variety of often debilitating symptoms. This can range from having vision or coordination loss to completely losing the ability to walk or being otherwise paralyzed. Despite these incapacitating symptoms, current medications cost thousands of dollars each year and often cause severe side effects. The effects of dietary supplements, as they're cheap, mostly risk-free, and can be easily acquired, are compared in this experiment. The 4 tested were chosen because in past studies, cholecalciferol has been found to increase the production of oligodendrocytes, methylcobalamin and lithium orotate have been found to improve the production of myelin basic protein, and docosahexaenoic acid has been found to, as a fatty acid, rebuild and strengthen myelin itself. And testing these supplements is all to answer the question, can any of these dietary supplements effectively regenerate the myelin sheath of zebrafish embryos? It was hypothesized that each supplement and each combination would be effective in regenerating myelin to some extent. This was later revised to exclude cholecalciferol, which actually had a negative effect on myelin regeneration. In this experiment, lipopolysaccharides, or LPS, which are demyelinating endotoxins, are used to simulate demyelination caused by MS on the model organism zebrafish embryos. Each supplement's concentration was decided through past research, which showed the optimal dose for ensuring some effect on the embryos without risking overdose and death. The LPS concentration was found through experimenting, where 4 groups of embryos were given various doses, and the one that had the greatest effect, characterized by deformities without causing mass death, was used. The independent variables are the treatments, while the dependent variables are myelin basic protein, swimming frequencies, tumor necrosis factor alpha, and physical deformities. There are also control groups, the negative only receiving LPS, and the positive only E3 medium, which all fish are kept in. At 3 days post-fertilization, the embryos are transferred into 6 well plates with LPS, and 4 days post-fertilization, they're transferred into 6 well plates with one of the treatments. For immunohistochemistry, the zebrafish were first transferred into vials of paraformaldehyde where they were fixed overnight at 4 degrees Celsius. They were then washed in PBST2 and incubated in primary antibody overnight at room temperature, and then they were washed again, and then incubated in secondary antibody overnight at room temperature, and then washed for the last time. For the ELISA, samples were prepared by transferring groups into 1.5 ml tubes where they were homogenized and centrifuged before being pipetted, along with standards, into a well plate and mixed, then analyzed with a microplate reader. Deformities and swimming were both just monitored through a microscope, and the swimming also involved taking videos of the fish for later review. The swimming frequency results, which measured groups' relative franticity, showed that methylcobalamin and docosahexaenoic acid were most effective in maintaining neurological health, while cholecalciferol and the combination of lithium orotate and methylcobalamin were the least effective. In terms of deformities, which include things like yolk sac edemas, bent spines, or pericardial edemas, lithium orotate was by far the most effective. The combination of lithium orotate and an unknown concentration of docosahexaenoic acid due to contamination was also effective, while cholecalciferol was not only ineffective but actually led to more deformities. The ELISA for TNF-alpha, which as an inflammatory cytokine can lead to demyelination, thus its reduction can lead to healthier myelin sheaths, showed that the combination of docosahexaenoic acid and lithium orotate and the combination of docosahexaenoic acid and methylcobalamin are both very effective, while each supplement on its own is not, except docosahexaenoic acid. However, though these results were confirmed to be statistically significant, the difference between the negative and positive control groups are not, implying that something probably happened to skew at least some of the results. This can also potentially be accredited to the multifaceted role of TNF-alpha in myelination. Immunohistochemistry for myelin basic protein was supposed to visibly, directly model supplement effects on myelin regeneration, and it did. However, there were 2 main problems with this test. First, to quantify the myelin present, a color selector tool was going to be used to find the percentage of each fish highlighted by MBP. However, the camera glare in the photos presented the same color as MBP, eliminating this method of quantification. This didn't matter, though, because the embryos this week had an atypical developmental occurrence where they didn't come out of their embryonic sacs until 6 or even 7 days post-fertilization, whereas they usually do by 3 or maybe 4. This means that they likely could not properly absorb the LPS nor the treatments while still in their sacs, invalidating the results of this test, and due to time restraints, it couldn't be repeated. All of these tests eventually lead to the conclusion that the combination of docosahexaenoic acid and lithium orotate, the combination of docosahexaenoic acid and methylcobalamin, and lithium orotate are the most effective supplements in myelin regeneration. Since zebrafish embryos have myelin biology that's homologous to humans, these effective supplements serve as potential treatments for people with MS. In the future, monitoring myelin through oligodendrocytes as a model organism would allow for more detailed review of myelin generation. Additionally, exploring different supplements that were not tested in this experiment has the potential to find alternatives that yield even better results. Thank you.